Hi, my name is Allison Wu, and I'm presenting on predicting breast cancer risk with 23andMe genetic testing. So, first, some background. What is direct-to-consumer genetic testing? Well, it uses saliva samples to provide information about one's ancestry, disease predisposition, disease carrier status, predicted drug metabolism, and other non-disease related types. This type of testing can be extremely beneficial as one can learn about their medical history, and it's also a more accessible form of genetic testing. It should be noted, though, that the FDA prohibits companies from providing diagnostic information to their consumers. But what are some of these companies? Well, you may have heard of 23andMe, MyHeritage, Ancestry.com, Family Tree DNA, or others. 23andMe specifically has over 14 million consumers using their test worldwide. This presentation also focuses specifically on breast cancer. The American Cancer Society predicts that in 2024, 310,000 women will be diagnosed with an invasive form of breast cancer. Of these 310,000 women, 42,000 of them will unfortunately die. But how do we determine breast cancer risk? Well, there are two genes, BRCA1 and BRCA2, which are responsible for repairing DNA damage to prevent cancerous growth in breast tissue. But mutations can arise in these genes, affecting the function of them and lead to increased breast cancer risk. Currently, we have identified over 4,000 BRCA1-2 variants. Let's say you get tested with 23andMe, though, and you get your breast cancer report. You may read you have 0 of 44 genetic variants tested, which may give you a sense of relief. But you might also see you have a greatly increased risk of developing breast and ovarian cancer, and that might elicit emotions of fear, stress, or even sadness. But should you believe these results, and how accurate are they? Well, let's first look at accuracy. So 23andMe reports on their own accuracy, saying they have rigorous standards for choosing genetic variants and genotyping protocols. The variants that they choose must be supported by multiple clinical studies, and that when genotyping in other lab conditions, they have greater than 99% reproducibility and accuracy. But on their website, under warnings and limitations, they also talk about how these types of tests are not a substitute and that the results should be confirmed by an independent genetic test. So I looked for some studies doing this, and I found a 2018 journal article published in Genetics and Medicine, and they confirmed uh, reported variants with direct-to-consumer genetic testing with clinical laboratory testing at Ambry Genetics. And their study had 49 patients, which were predominantly female and Caucasian. And for these patients, when looking at a variety of variants, including breast cancer, but also like Ehlers-Danlos syndrome, they found an alarmingly high false positive rate of 40%, which means that only 60% of the variants reported by these genetic tests were be confirmed with the laboratory testing. For breast cancer specifically, there was still a fairly high false positive rate with 32%. 68% of the variants were confirmed. So it seems that 23andMe actually has fairly low accuracy. But what about their uncertainty? What is the likelihood of developing breast cancer? Let's say a BRCA1-2 variant is found. Well, the CDC reports that 50% of women will develop breast cancer by the time they're 70 years old. And this doesn't even take into account family history or environmental factors, which can greatly affect risk. But testing does inform preventative measures like perhaps getting a prophylactic mastectomy or other treatments. And what about the likelihood of developing breast cancer if no variant is found? Well, a non-peer-reviewed study presented at the American College of Medical Genetics and Genomics annual meeting found that a high number of mutations were gone undetected. 90% of 100,000 study participants would be missed with the 23andMe BRCA1-2 test. But why is that? Well, 23andMe has an extremely limited scope. They only test for 44 BRCA1-2 variants. But remember, there's over 4,000 of them out there. And BRCA1-2 variants are not the only types of mutations that lead to breast cancer risk. But for these variants, three of them are present in 1 in 40 individuals of Ashkenazi descent. So 23andMe's test is not only limited in number of variants, but also the type of variants that are present in a diverse group of individuals. And another thing to note is that 23andMe utilizes genotyping, which is kind of like picking and choosing a variety of letters in a book to read or words to read, whereas sequencing is looking at every single letter or word and getting a full picture of what is there in your genome. So, of course, within genotyping, there's going to be more uncertainty with predicting breast cancer risk because you're not getting that full picture. So there's fairly high uncertainty and low accuracy with 23andMe in predicting breast cancer risk. But how aware is the public? Well, 23andMe does actually a fairly good job communicating with the public, in my opinion. They're aware of the uncertainty linked to their tests, and they encourage users to learn more about their results. As you can see here on the right, this is actually what I see when I log into my 23andMe portal. After scrolling, looking through my breast cancer results, you can see that there's this tab to learn more. And there are multiple resources here that are written in simpler terms that are able to be digested by someone who's not as well versed in medical literature. So, should we use these direct-to-consumer genetic tests, and how should they be used? Well, I certainly believe that these tests can provide useful information, but we should be aware of the accuracy and uncertainty of these tests, like which variants are being looked at and how many of them. We should also seek guidance from healthcare professionals about results before taking any medical action. We should remember that a positive or negative result does not confirm one's development of breast cancer in the future. There are many factors that can affect breast cancer risk. So I've taken the test, but will you? Thanks for listening. I hope you enjoyed this presentation, and here are my references.